there's this thing called uh, the blind source separation, which is, if you take a convolved signal C, can you deconvolve it back into A and B? It's kind of like a PCA or ICA kind of thing. And I think he was actually able to feed these. Well, he initially trained the neurons to recognize signal A and signal B separately. And then what he did was he then played back, I think, signal C, which was a convolution of A and B. And to his surprise, I think the neurons that had been trained on on A, and then the ones that were B were individually like responding to the actual like mixed convolved signal or something of that. So I can't remember exactly details of that. So anyway, that happened. And then I was thinking about it at the time and said, wait a minute, if you could actually do that, right? I guess because it's a computational problem. What other computational problems could you solve with these biological systems? And if you could like do that and scale it up, could you end up with a biological computer? So that was, I guess, where the idea of Cortical Labs was born, sometime in mid 20 to late 2018. And we were, brainstorming ideas of what could we do, what do we want to do. And then the inspiration, I guess, came from the early DeepMind experiments where they were building reinforcement learning agents to play the Atari games. And lo and behold, what did they do to start? They started with Pong. So I said, wouldn't it be cool if we could also get these neurons to play Pong? And that was where the birth of the idea of this paper came from. And so, yeah, we spent the last 3 years growing a ton of these neurons and working on this. And we actually got a preprint out last year. So this publication that you'll be reading is actually more of last year, well, 2 years of a year and a half previously of research work. We're doing some really exciting stuff right now, but maybe I'll open the floor, or if anyone has any questions, they can feel free to ask.
I have lots of,
No?
but I'm sure that people have questions. But if not, I can go first. Questions to you. Okay. And then yeah, we have a spreadsheet that we kind of prepared ahead of time. So some of these are mine and some of them are not.
Yep.
So I guess what surprised you the most in your career so far?
What surprised me most? That's a hard question. I think it's I guess this whole startup has actually been very surprising. Honestly speaking, actually, it's one where we didn't expect to actually get all the way to this point. It is, I think it was extremely risky and daring for us to actually do this experiment, primarily because there weren't any other experiments that we felt that had actually done the same thing, apart from the Isubura paper, which is the closest one. There have been previous experiments done by Steve Potter from Georgia Tech, and a whole bunch of other people. There's a whole like Twitter storm moment of like, why didn't you cite it, blah, blah, blah. But they were all very small scale. They were not very reproducible. They were missing details of like, where did the cells come from? What are the characteristics of the cells? And, things were, n sample size of 2 with maybe an hour worth of like data recordings. And at that point in time, you're like, well, can I reliably use this data to then base our experiments on? And the answer was actually no. So a lot of this stuff, some people always say, oh, you gain inspiration from X and you didn't cite or whatever, was actually clean room implementation, because we thought that there was a lot of stuff that was missing, and that we just had to just think of it for ourselves. So I think that's probably the biggest surprise, that we've actually managed to get to where we are here. No chance to a bit of luck as well, I guess. It was quite lucky that we were able to sort of come up with the schema that worked with the encoding decoding thing. And I think this was one of the hardest things for us as well, because as if you're a computer scientist or anyone, you're trying to outsmart the system and say, hey, let's build an algorithm to try to like linearly map inputs and outputs and all that stuff. And if you read the paper, actually you don't see it here, but in the preprint, we actually talk a little bit about this EXP3 algorithm. And the problem with EXP3 was they got a whole bunch of people extremely confused, because they were like saying, wait, so is this an algorithm that's actually interpreting this and so forth, when in fact EXP3, I'm not sure if you guys understand, know much about it, but it's more of an algorithm to solve the multi-arm banded problem, right? How do you optimize configuration scheme, in a finite space in the shortest amount of time? And so we said, you know what, like we'll put it in the other section, because we did initially use EXP3 to try to figure out which was the best schema, but it was too complicated. And what happened was we realized that it was bad for the neurons, because the neurons didn't actually have a ground-based model truth to start off with. And you had this thing that was dynamically switching it. So you almost end up having a dual attractor problem, right? Where one would be moving, and it would then try to like reduce its sort of gradients on one side. But while that was happening, this one was also like changing its parameters. So you never end up settling into some sort of global minima. So we just made the decision, said let's just fix the configurations and not touch them. If the neurons are truly plastic, they will reconfigure and reform to that specification. And if they don't, then well, we'd run out of money and time. So it was a bit of a gamble, and I'm quite thankful that it kind of paid off.
I got you. So, okay, so to clarify, so you have the struggle was sort of like there's a way to encode the game of Pong into like stimulus for the neurons, and then there's a way to decode the what you measure from the neurons into input to the game of Pong, right?
Correct. Yeah. I mean,
those are
yeah.
those encodings and decodings are those, you're saying the EXP algorithm is that, and then you said it was multi-arm bandit, is that like reinforcement learning or
no, it was more of trying to figure out the schema of things, like which ones do you use, right? Actually, let me just show you. It might be easier this way. I'm just gonna share. Oh, can I share screen, please, host?
Yeah, sorry.
I'm not sure you guys have seen this, but you can actually just check it on your own computer or phone or tablet. It's just spikestream.corticallabs.com. It's a visualization that I built to help visualize the activity. So you might have seen this. This has been going around making the rounds. So I'm just going to refresh this, and I'm going to show you. So a lot of people don't understand what this is. So I'm just going to walk you through. So this is actually a visual representation on the surface of the CMOS MEA chip. So the CMOS MEA chip that we use is a Max1 MEA developed by Maxwell Biosystems. And so this plane here is roughly the size of pinky nail. So copper metal oxide electrodes. And there are 22,000 of them in this arrangement. So what we then say here is this is electrode number 0. This is electrode. I'm just going to zoom in a bit closer. So this is 0, and then it goes to 1, 2, and so on and so forth, right? But then, so you end up with this sort of linear view, but then you also end up with this XY positioning. So what we then do is we say, okay, how do we encode this information of where the ball is to the paddle, right? Because we need to feed, if you think of it, the neurons as an agent of sorts. Like, what is the information that they're going to need to be able to play this game? And how do we do it within the limitations of the capabilities of the hardware? And so the Maxwell Biosystems setup has only really 8 stimulus units. And when I talk about stimulus, I'm talking about like the chip giving stimulus to the neurons and not vice versa. So it only has 8 stimulus units, but it has a read capability of 1024. And the reason why they did that was this was mostly for drug development assays and so forth. And I think STIM was just a last minute, thing that they weren't really interested in, but they just threw it in, for shits and giggles. So you now have this problem of how do you take information from a game world, from some sort of structured information landscape, and feed it into the cells? And then how do you take the information from the cells and feed it back into the structured information landscape? And so we had lots of different ideas, but the one that came to mind was this, which is a very simple representation. And it's, mind you, it works, but it's not particularly a very good encoding scheme. there probably are way better schemes to actually do this, right? And it's just one that visually we understand, it works, but not optimal. And the way we've done it, if you read the paper as well, is that we have, so this is, all of these blue units are units that have been activated. So if you add them all up, there'll be about 1024 of these regions. What we then do here is we encode information. So you can't really see it because we don't really put it into this visualizer, but you can imagine there are 8 stimulus sites in this top region. This is the data input. So what we then say is, well, if the game of Pong is happening here, we encode information of where the position of the ball is to this paddle. Is it, closer to the center? Is it bottom or the top? In sort of like 8 different regions. So that's what we call place coding, right? It's very similar to topological place mapping that you have in like the auditory cortex and so forth. And then what we then say is that this distance, you can think of it like the x-axis distance, is encoded as a rate encoding scheme. So the closer the ball gets to the paddle, the higher the frequency or intensity of the stimulus. And the reason why you don't see this here is because we blind this system to our own stimulus, right? Because we don't really want to be picking it up. And so this is how you input information in. And then what we then say here is these 2 regions here are the motor outputs. So these regions here control the paddle going up and down. How do you do it? Like, there were a lot of different configurations, right? This is the one that was very difficult to figure out because there were so many ways to quite literally skin the cat for this one. So we use EXP3 to say, hey, should we do it as this entire chunk to be like up and this entire chunk to be down? Do we make this one down or an up, or how do we do this, right? So if you think about that, that's like a 2 to the power of 3 or 4 different combinatorials. It turns out what actually was the best result was having a split config. So you'll see in the schema in the paper. We don't really see it on this one here, but this is actually, I think, up or down, one or the other. But it's actually it's also got the down immediately up to it. So if this is up, this is down, this is up, and this is down. And what this does is create this natural phenomenon called lateral inhibition. And you do need lateral inhibition for these things to really work. Same thing as you probably like studied in like the visual cortex, right? Where the retina cells are looking for edges, right? So one will be a inhibitory system because it's looking at, say, a white screen, but then the moment that a black line comes along, that becomes an activation. So you need to have this contrast. Turns out, yeah, we found out that you needed this for these neurons as well. So what we then do is we say, okay, we're going to feed this information into this region here. We're going to wait a short period of time, and then we're going to sample in that period to say, hey, cells, do you want to push the pedal up or do you want to push the pedal down? And that's, if you see this game visualizer, is essentially what the neurons are doing. They are pushing this pedal up or down based on how much activity that they are having in these so-called motor regions. So you feed it back and forth, and within 5 minutes, they actually start to improve their performance. So they're moving this pedal back and forth more often to hit the ball than not. I think one of the crucial important things that we needed to do for this was also figure out what was the way to push these neurons down a particular pathway. And one idea was to use Professor Carl Friston's theory, the free energy principle, to say, well, if all biological systems, all intelligent biological systems are in existence because they want to minimize the amount of information entropy, the information surprise, which is the delta, or you can think of it like the KL divergence of sorts, between its internal model representation of the world and the observed representation of the world, then if you were to say increase the amount of information entropy of sensations coming in, that would then push them away from doing that action and instead favor getting information that is more predictable. And so using a information entropy scheme, we were able to influence the neurons to move the paddle. And so every time the neurons move the paddle and it hits it, we give it a predictable stimulus. You can think of it like either a topological one, so like a straight line that's quite predictable, or you can think of it as a rate coding scheme, which is like a sine wave, right? Because from one period, you can predict n number of periods going forward. So and if they miss, we just randomly give them information. So that's essentially, I guess, in a nutshell, how this system works. Any questions, comments?
Yeah,
Yep.
we got a couple. Vernon first. Yeah.
So I'm a little curious. You're talking about how information entropy, the system doesn't actually, the neurons don't actually like the fact that you're giving them like unknown stimulus. But in temporal difference learning, like most like biological systems actually like learn from the unknown stimulus. So how was it that you're able to give them like almost like useless or like overload of information, and they didn't necessarily adapt to that, but instead they adapted kind of in a different way by going away from learning, like in a traditional sense?
Yeah, I think it's I think the thing what we did was so again we don't really know exactly why. We've essentially used this as a principle and we've tried to apply it. I think this is different from the temporal difference learning, in the sense that this is a if you think of it, we have a natural Markovian blanket that happens between the cells in this dish, which what they are trying to do is recreate the model of this game world, this Pong game, right? Because if you think about Pong, it has certain rules. For instance, if a ball hits the back wall, it will bounce at a 90-degree angle, and it will continuously go in that vector trajectory until it hits the paddle, and so forth. So by having that structured information landscape as a game, right, and you have these neurons on one end, you have this natural like Markovian blanket that happens between the neurons, which we think are trying to create some model of what this observed structured information landscape is for the game, and trying to improve its performance so that it can predict in the next game, because every time it misses it, we scramble and then we reset the trajectory to try and predict the better, so it doesn't get that. As to how or why it conflicts with the sort of schema that you're talking about, we're not quite sure yet.
Do you think maybe it's because like in TD learning, it's based on dopaminergic neurons specifically, and maybe that's why it differs?
It could be. I mean, like, I guess maybe in temporal difference, there is an element of Hebbian plasticity that happens with that, right? Because if there is a coincidental firing, which is an unlikely event, but it happens often, then it's something that these neurons ought to learn, kind of thing. So I think this was something that we kind of didn't really want to do too much, which was we didn't want too much Hebbian learning to happen, because we didn't want it to just memorize it. We actually wanted it to learn how to actually control this paddle.
Yegor, did you have a question?
Yes. So my question essentially comes down to, do you think that there would be an interesting research direction to try to model this computationally? Because what we could do is it's not that impossible to create a simplified model of a neuron that more or less behaves similarly electrically. I don't believe we fully have complete understanding of everything that goes on, but we could probably simulate on, like, for the most part, the order of magnitude number of neurons that are being used here. Do you think you can try and create a simulated version that learns in exactly the same way and extrapolate from that sort of simplified algorithms to potentially improve our more traditional reinforcement learning techniques? Because ultimately, oh yeah, go ahead.
Possibly. I mean, so people have asked this question previously, and I think it really boils down to the question of do we know the exact model of what these neurons are doing, and how they're actually computing, and also do we have the hardware to do it, right? The answer, I think, for this one would be my from what I've observed and from the data that's come through, is that it's going to be very difficult. It's doable, but very difficult, because let me give you a reason why. So in biological neurons, every neuron has slightly different activation thresholds. And this activation threshold is dynamic. It changes based on, as you've said, dopaminergic systems will reduce the threshold for firing. You have inhibitory, excitatory systems. It's very complex. And in a computational system, right, the nonlinearity is fixed. Your ReLU is fixed, for instance, or your tanh or whatever. And that allows massive parallel computing, right? If you have a fixed function, then all you have to do is, data in, process data out, and you can use a GPU for that. If you don't have a fixed function, right? If your ReLU and whatever is changing per neuron per time step, you no longer can use a GPU to compute for this stuff. You'll now have to start using a CPU. Then you run through a loop. So when you do your for loops and you're trying to do this, it takes a very long time to sort of calculate them. And so that's why I'm saying it's not impossible. I just think it's very difficult. And we have about 800,000 to 1 million neurons in this. So can you imagine running 800,000 simulations on a CPU loop, as opposed to churning it through a GPU for this kind of stuff?
So essentially, we first have to contact Google and have them do it. Yeah.
Possibly, but even Google, I guess, have spent a lot of resources on their TPUs, their tensor processing units, right? And if you think about where we are today with artificial intelligence and reinforcement learning, none of this would be possible without the fact that we are able to crunch through large amounts of vector and matrix multiplications using GPUs and TPUs. We still have very poor very slow systems if we have to do CPU based learning. In fact, nobody does. I don't think anyone does CPU based learning anymore, because of just how slow it is. But yeah, it's one of those things that I think we don't quite know yet, and there's a significant amount of uncertainty of how these neurons work. If you think about it, there's in the spiking neural network space, right? There are I think 5 or 6 different models, these Sikovich, leaky integrated fires and so forth that are proposed, and no one quite knows exactly which one is the best one to represent actual biological spiking neurons.
So on that note, I guess given the sort of what you're trying to make a biological computer, so what are the advantages that, I mean, we already kind of covered some of them, in that they can do these sort of analog computations before doing something, before kind of turning it into a digital computation with firing neurons. So what do you see the future of computing, and where do you see biological computing in that?
Yeah, so I think the interesting thing that I've observed and been thinking about lately as well has been the concept of time. And time is something that isn't actually really discussed much in the, I guess, machine learning, reinforcement learning space. I remember I was talking once to a ML researcher, and I was talking about, how do we model this time? He's like, oh, just put it through a CNN. I was like, what do you mean? It's like, yeah, it's a strip. Just, put it through a CNN and I'll just look through it, kind of thing. But I was like, yeah, but, shouldn't the previous data frame inform the next data frames if you think about, how we process things in a Bayesian way? And I think they were just puzzled, because they're like, well, the CNN works really well. Why wouldn't you use it? And to the point with that is that in digital systems, particularly in machine learning, or the current stuff that we have, you collect a huge amount of data first, and then you're learning on that. And there's a whole bunch of things with meta and online learning and so forth that's happening at the moment. But in the real world, you don't have that luxury and capability. You got to build the model. You got to have your priors set, and then update them as you observe the world, kind of thing. So my feeling and my understanding is that these biological systems are somehow doing computations that are extraordinarily data efficient, the sample sizes, the input samples that they're getting are extremely sparse. They're very low in terms of the amount of data that we're receiving, right? And that's because we're running at analog clock speeds that you and I all agree at, right? So for instance, we can't speed up time, or when we watch this game of Pong on the screen, we can't speed. I mean, I guess you can record it and speed it up, but as you're watching it now, you can't really speed the game up. And so you get given the number of frames that nature and the world wants to give you, and you can't ask for any more, and you have to do something with it. So I think that's like one of the key things, which is the sample efficiency. It's going to be really important for like real-time applications, such as say robotics, right? Where your robot doesn't have time to sit there and collect the data and then like build the model and action on it, particularly if it's in a scenario that is unfamiliar, that it's new, that it has to like adapt to. And then secondly, things like cybersecurity challenges or issues where time is of the essence, right? You don't have time to sit there and collect data while your system is being penetrated and it's being wrecked as a havoc, because at the end of the day, the worst things are zero-day exploits where you don't know what's happening, it's completely new, and you have to dynamically deal with it. So I think those are one of what, like, data efficiency is one of it. The other one is going to be power efficiency. So I think I did a rough back on the envelope calculation, and it's not even a very good calculation because I assumed there was 100% sort of aerobic respiration, combustion, and so forth, right? So I just said, how much sugar are we putting into the cell culture media? How much are we consuming, and how much are we replacing them? And even if you did a very inefficient calculation, it blows my mind that these neurons are only sipping about 10 to the minus 4 watts of energy, and are able to sort of do this kind of task. That's even less than the actual like machinery that we use to keep these cells alive. So it's incredible if you think about the amount of energy that these things are sipping. And if you take a step back and you abstract it away, it completely makes sense, because in the natural world, one of the scarcest resources available are calories, right? And so neural biological systems have adapted to use the minimal amount of energy required in order to get around, not get killed, not get eaten, and reproduce. So I think that's one of the things that we're hopeful that we can use to create more efficient systems.
Yeah. Do you think like the ability for neurons to restructure the thing that is doing the computation, like as they compute, like you were saying about convolutional neural networks, where shouldn't the output be the input for the next one? I mean, that's sort of inherent with these types of things. Do you think that is fundamentally different from the ways that we compute with machine learning now? And that we could
I think there is a significant amount of difference. I mean, we've tried to do there's some work on recurrent neural networks, Boltzmann machines, and so forth. And I think those are closer to actually closer to what is actually happening in biological systems. I know Jan LeCun's looking into a whole bunch of these energy-based systems and RBMs and so forth, which I think is the right path. Just my 2 cents, that's all.
Yeah. I have more questions, but does anybody else have a question they want to ask? I'm gonna leave that for a little bit.
Actually, let me see if I can pull it up here. This is actually not available yet, but we will be putting it up on archive soon. And the reason why it's not available yet is because it's going through peer review at the moment. Where is it? Here we go. So we actually have a paper right now trying to look into this, which is a somewhat of a weird comparison, but we're trying to compare reinforcement learning systems to the biological the biological game. So what we did was we took DQN, A2C, and PPO as algorithms. We fed it we use the same game engine essentially as our biological game to try and play the Pong game and use the same rules and so forth. And what we then did was we said, yeah, everyone's saying, yeah, you can train these reinforcement learning agents to like, be superhuman level within like, 10 minutes of training and so forth. But that's it because you're accelerating time, right? You're doing batch sampling sizes of like 10,000 episodes per second or whatever. And so what we then said, okay, we're going to play this game, but we're going to cap the number of episodes that these reinforcement learning agents get so that they see only the same number of frames that the biological network has. So most of our games have been playing for about 20 minutes. The game runs at 100 frames per second. So 100 times 60 by, 20 or something like that. Yeah, so that's the total number of frames that we had for our biological system. We capped it. We capped the reinforcement learning agents as well for that. And what's really interesting is that you see what we've done is an average of all the gameplay that I'm just going to zoom in here. Average hits per rally, which is the kind of one of the metrics that we use. The biological systems actually learn faster, or they learn faster given the same amount of samples than A2C, PPO, which is really weird. PPO seems to go backwards, and I guess DQN, which is a really slow learner. So this is something that we are very keen on exploring further to try and understand, how does this system sort of like work, and how can we actually like use this to complement the current reinforcement learning AI space.
So just to clarify, the two lines that are different from the rest, the orange and blue lines, were just biological systems.
Oh, sorry. Let me just put it back. Yeah.
To clarify, the blue and orange lines there are the biological system. Both of them are.
Yep. Yep. Sorry. MCC stands for mouse cortical neurons. HCC, human cortical cells.
Okay. Thank you.
Yeah.
So, okay, so if these are like more sample efficient than our current algorithms and more power efficient, do you see a future where there's like biological chips in computers? Or, and then my other question is like, so having worked at an organoid lab, we did kidneys, but cerebral organoids are by far the biggest that people have been able to grow, from my understanding. And it's like, that's a little scary. Like, I know that they have the issues with vascularization, so the little brain eventually just gets too big to feed itself, and then it dies. But that seems surmountable, and I know people are working on it. And then that's a little scary as well.
Yep.
How do you feel about that?
Well, I think so. You gotta do it for a reason, and it has to be a good reason. Kind of like the guys who, I think Nature just published also about the coinciding with our paper with the human organoid implanted into a mouse thing.
Oh yeah.
And I was like, that's a bit, I get weirded out more so by chimeras than actual, oh my God, these organoids are going to be sentient. Because if they are sentient and then you're putting it into a mouse that you have, what do you have, like a human mouse, kind of weird chimera. So anyway, but they had good reason for that, which they were trying to experiment and see if you could actually grow organoids, could you then reimplant them back into human or host, whatever, and maybe those organoids would then resynapse and hopefully restore functionality. So if you had a stroke and you had a portion of the brain that got liquefied because of the ischemia that happened, could you just put it back and then have that sort of like fill in the space? Back to the question of like, I guess, is it scary if these things become more complex and they start having better or having weirder sort of like responses? I think it certainly is the case, and something that we definitely have to watch for. I'm not too worried about it, kind of thing. Kind of like what Andrew Ng talks about, like AI is getting sentient and conscious. It's like, overpopulation on Mars, we're not quite there yet. And I think that's something, that we all have to sort of think about, keep in the back of our minds. And if we do see something, we then have to start thinking about it, right? And if we do it, there has to be good reason. For instance, like, we can't say replicate dementia-like behavior in a two-dimensional neural culture. So then we'll have to grow the organoids. But that's for a good reason, right? Because we're trying to figure out a way of saying, creating cures for dementia. So that outweighs the risk of that. And in fact, it might even be better than, giving mice dementia, through some sort of CRISPR modification and then doing some testing and killing them, right? Because a mouse is technically a more complete, more I would say even conscious organism than say these organoids in a dish. The only difference here is that they have 23 chromosomes and are more similar to us genomically than a mouse. But the thing here is that it's not just the type of neuron, right? It's not just that they are human origin and so forth, but it's also the organization and the structure. Are they more are they more organized like a human brain, or are they more organized like a mouse, or are they just a flat thing, or are they just like a blob, right? I think these are important questions that we all have to think about as we go forward.
Yeah. So, do you think this is something that would, it seems like it is helping us understand how to engineer consciousness. I mean, maybe not your work in particular with playing Pong, but in general, learning how to build these like cerebral organoids. Maybe you guys do work sort of on this. So if we were to sort of learn how to engineer consciousness, or maybe you don't think that's possible, I'm not sure, but then sort of what are the consequences of that? And is that something we should do, or yeah?
I mean, that's a good question. Some people may say it's a good thing to do, right? I think it really depends on the context. Why are we doing it? What's the reason for it? If you want to create consciousness because you can, then that's probably not a good enough reason. But for instance, if you said, I wanted to create consciousness because I want to study how coma works, right? Because you get consciousness in a dish and you knock it out, and then can you revive it back? Because you're trying to create a cure for people who are in comatose states. Can you wake them up? Can you do all these things? Or is a comatose like organoid similar to a person in that case, right? And so on and so forth. So there has to be good reasons behind that. And I think there are reasons where studying consciousness in a dish in a controlled fashion is actually beneficial for humanity in a greater sense, right? There may be other reasons behind that. I think the possibilities are quite open. It just needs to be discussed, right? As a community, as people in today's age, right? Any other science, a healthy debate and discussion about, is this ethical? Is this something that we should be doing? Is really important.
Yeah, gotcha.
So I have a question that's more related back to like RL and deep learning. So
Yep.
I have my opinions on this, but I'm curious what you think about. So do you think your research kind of shows that there's something kind of fundamentally missing from contemporary RL and deep learning methods in their extreme lack of sample efficiency and just general like inability to do cool things in certain contexts? Or is it more along the lines of what you talked about before, where it's just like these kinds of deep learning methods just process data in a sort of just the way they do things in batches, where it all relies on parallelism rather than processing that? I know we can't actually simulate it directly using contemporary technology, but do you think the techniques themselves have something fundamentally missing? That's kind of my question.
Really good question. And so I think of it as two different. I think of it as so I think to answer your point, I think reinforcement learning has done a really good job so far. And the way I describe it is that reinforcement learning is like a plane, right? It uses a great amount of like energy to work, and when it does, it's pretty cool, right? And it's the same thing with a plane. A plane consumes bucket loads of kerosene just to like take off the ground and can keep flying, and it can do a lot of crazy like heavy lifting and supersonic speed and so forth. But on the flip side, you have a fly that gets around, doesn't use as much energy, but it's pretty damn efficient, and it still flies. So you now have these two different models, right? Now you have a model that can fly, that we have mechanistic models for, and you have one that's also a biologically inspired one, which uses much less energy to get around, kind of thing. And I think it's the same thing to intelligence. Reinforcement learning systems, if you subscribe to the idea that the world is full of structured information, right? In nature, we see a tree. There's in the tree there's an edge, and it's against the backdrop and so forth, right? It's not random noise, random information. There is structure to the world, and the work that's coming out from reinforcement learning, and particularly also in transformer land, right? Like all those DALL-E 2 GPT-3 things. What it's showing us is that what these neural models are doing is that they are learning high dimensional representations of the structured information landscape that we feed the information in. In this case, ImageNet, right? Or I don't know what's the ImageNet equivalent for NLP?
GPT-3.
Wikipedia, I guess. So we're feeding all this information in, and it's clustering information, right, in this high dimensional space. And then what the transformer networks or the reinforcement learning systems are doing is they're drawing boundaries, and they're saying, okay, well, there seems to be this high dimensional cluster of these things, and they seem to have this labeled dog. Maybe the cat one is down here, kind of thing. And that's essentially what our RL systems are doing. I think the biological systems are also doing something very similar. And if you subscribe to this free energy principle model, they are doing it as well, because they're trying to model the world, except we're doing it in a fashion that we don't quite understand from a digital form. It is doing it somehow in an analog fashion, in a much faster sort of loop. And if the way, so to highlight this, right, it's kind of like some people talk about like, oh, how do we know about this free energy principle, and how does it apply? The way I would say that it applies, that you can sort of like think about when you get it, is whenever you get a new pair of glasses, or when you get a new pair of watch, you notice that like there's something weird about this for the first 1 or 2 days, and then after a while you just don't notice it. It's just there. The glasses just are there. You don't even notice you're wearing it, or the watch. And that is essentially a way of the brain learning, right? It's creating a model of what the world is continuously at any one moment in time. But once it's figured it out, it's like, you know what? I'm not going to spend any more computation about this. I know what this new watch feels like. I'm not even going to like compute that. So I have a feeling that that's what the biological systems are actually doing. And that's how they're much faster, because they're not computing everything else. They're just storing it as weights or as distributions somehow in the synapses. And then they're just processing new stuff along the way. And we have a pretty good model of how the world works from 10, 20 years of life experience. And actually, going back to the point, this is an aside as well, the question about consciousness as well is, I think people also seem to think that consciousness is an inherent human property, but I kind of wonder if that's really the case, because how many of us remember being born, right? Nobody remembers being born. In fact, the earliest memory that we have probably like is when we're like 3 or 4, kind of thing. And if we don't have memories and recollections of things that happened before then, does that mean we were really conscious? Because consciousness kind of really needs this kind of, I'm self-aware and I'm building memories of that point in time. So does consciousness really exist in very young children and adults, right? Does it emerge from the later stage where we then start having things like, what is it, the theory of mind kind of thing, and self kind of thing, which is an evolution that happens a bit later on in a child's development. Sorry, that's gone a bit longer for the question.
Very good answer, thank you.
Yeah, I agree. That was good.
Yeah. Can I ask another real quick question?
Sure.
Actually, I phrase this. So one thing that has recently kind of interested me.
I can't hear you. Is
Speak up.
something going on with my audio?
Can you hear me?
Hello.
Was it muted?
Hello.
You guys hear me? I can't hear you though.
We can hear you.
No, still can't hear you.
We can hear you. I think it's your headphones, bro. I don't know how to tell you that.
Oh wait. Okay, let me 1 moment. Try it
Yeah.
now. Oh, I can hear now. Sorry. Okay.
Okay.
My side.
Okay, you can hear?
Yep.
Okay, so I guess my.
Sorry if I didn't hear anyone. So if anyone was actually asking a question and I was just rambling, I apologize because I didn't hear a single thing for the
Yeah.
last 10 minutes, I think.
So I guess the extension of my question, this isn't really an extension, this is just a side tangent. But one thing that I think is interesting about, especially the work that you did, is compared to like deep learning methods, neurons operate purely locally. Like no neuron can tell what's happening. Like I don't know how big the chip is, but let's say it's like 3 centimeters away. Unless it has a synapse directly to there, it can't tell what's happening like through some chain of neurons. It can only tell what's happening around itself in some kind of cluster. And yet, despite that, all of these neurons must somehow be creating a like model of not necessarily the world as a whole, but their local environment of neurons. And
Bingo. Yes, that's correct. So for instance, we don't even know exactly like the effect of local field potentials are, right? Because as a neuron fires, right, it's generating voltage differentials, not only internally, but it's external space. And because of the external space, right? So you think of it like a gradient, like there's a voltage firing, and then you can detect it further down the way. But if there's another neuron on that side and it also fires, and then you now have like superposition of like electrical voltages, you end up with also a very weird, very interesting electrical landscape that is not of the neuron itself, but its local surrounds that we don't quite capture that in digital networks.
yeah, that's kind of the essence of what I wanted to get to. Thank you.
Yeah. Excellent point. Like, yeah, the local field potentials are something that we don't actually even like look for. And in fact, in our new system that we're working on, we actually have decided to actually not go down the path of having that high resolution, because we were like thinking about it saying, yeah, we've noticed that most of the actual voltages that we're picking up, while we can get like an action potential from an axon underneath, is actually more the local field potentials in that particular region that seem to matter more than anything else. It's almost like you can think of it as like the neurons are like voting, right? They're like, oh yes, we all want to go down this path, and if they all sort of fire in that same spot in that vicinity, it doesn't really matter if you're right underneath the neuron. You just need to detect the whole the actual local field potential of that cluster.
Dang, that's good.
Yeah. And you know, that's actually one of the, so you know, people, if you want to know, like I guess some of the stuff that we're working on in the future, one of the hardest things is actually this encoding decoding problem. Like how do you represent digital information for an analog system, right? I mean, it's kind of easy for a BCI to do that, right? You stick a BCI in, you tell somebody to move the hand up and down many times, and then you record the spikes, and then you fit some sort of model to it. But how do you go the other way back, right? You know, what is a dog picture or a dog image or a number 7 into a neural system? We don't know that.
Yeah, you need like a whole visual cortex to do that.
Exactly, right? You need a whole visual cortex, but then from the visual cortex, you also have pre-processing that really happens in the retina and along the way. And so then it becomes a question of like, how is that piece of information going to be transmitted? And in time as well, right? Because all these systems are all spiking nets. So spiking nets use time as the extra dimensionality in order to reduce the amount of data that's required to be transferred. So, and this, I think this is the craziest thing. And the one thing that I think about spiking networks a lot is the fact that it's so energy efficient, because a neuron isn't going to spike if there's nothing happening, right? It needs contrast. It needs something to like fly across the thing in order to go, oh, there's a dark spot right there. I'm going to fire and I'm going to transmit it. right? And if there's nothing, like if there's this white wall behind me, your brain, your networks aren't actually even spiking, because they just don't need to they don't need to send any information across if it's not changing.
Yeah.
I'm kind of done with my questions.
Okay, I have, unless anybody else has any more questions, I have one final one. Okay, it has been very cool to talk to you. And because I think that the work you're doing can impact the world quite a bit, I think it's important to ask you, what do you think 2040 will look like, and what do you want it to look like?
Oh, what do I 2040, how many years is away with that? That's like I guess 20, no, that's 18 years away. Well, I guess what I would really like this technology to do is I've been thinking a bit about this, and we've got some feedback from some people who have actually asked quite a lot for cures, like for Alzheimer's and Parkinson's to be developed and so forth. And it's interesting because we started out trying to build a biological computer, but now we started to think about it. It was like, so if you have a biological computer, and as you were asking about these applications, what are the ones that really matters, and so forth. And we internally in the team, we've been thinking about it. We're like, we could build a biological computer that could do some of this robotic stuff, but it's still going to take some time. But what is the more immediate benefits from this? And the work that we're doing with this alcohol study and also with the various drugs, I think actually has a much closer impact. And what I'd like to see by at least hopefully by 2040 is that a system like ours can be used to mass screen a whole population. All right, so we can say, right, well, with consent, of course, give us some of your blood, give us some of your skin cells. We'll grow stem cells from them, and we'll grow neurons, and then we'll run drug assays across them, and we'll give you back the profile, so that when you do get sick, your physician is then going to try, randomly try drug A and then work its way through. He's going to give you, or she's going to give you drug X, which is the one that is going to work best with the least amount of side effects the first time round. And that's going to alleviate a whole bunch of suffering. It's going to make, people more productive and happier and so forth. And also using that kind of system to screen compounds for like new drugs, to cure Alzheimer's, to cure Parkinson's and a whole bunch of these neurological conditions. Having said that, in order to actually do these things, we do need more complicated games. We do need more complicated computational environments, right? I think Pong is a nice start, but can we scale it up? Can it get the neurons to play Doom or whatever, right? Because that way, we will have a more robust system to test against these compounds. And so from doing that, the hope is that the computational side of things becomes a side effect. Kind of like how the way I think about it is like Amazon, right? Amazon initially started selling books, and they had a storefront. And in fact, Amazon.com is still a shop, right? But they built a whole bunch of backend stuff in order to power the shop, AWS. And it so happened that they said, you know what, we got all the spare capacity. Why don't we let the other developers use our infrastructure? And so we're taking the same approach as well, where we're developing our systems to, create in vitro cognitive models that we can use and test new compounds and drugs on. That is a platform to develop sort of mini AI games for these neurons. But could somebody take this technology and run along with it, build, more powerful cognitive systems, right? More complicated environments. Could they take this, put it into a robot? Could they stream information in from the internet, say a stock ticket price, or the price of Ethereum or Bitcoin, and get these things to learn how to trade for them, right? I think this is kind of the approach, and hope that by 2040 we'll actually have a whole bunch of people using these systems to compute and do the sort of AI tasks that we are trying to get our systems to do right now. That's kind of the hope, an ecosystem of developers and technologies that spring from this kind of base level technology.
Gotcha. So I need to start learning how to program in like organoid.
Pretty much. Well, we're working on it actually. So And please join the Reddit. We love having students and anyone come along to chat. We are actually building an IDE at the moment for Python and JavaScript. So you can use the APIs that we build to interact with the system. So, you can define what a motor region looks like. You can define what the input scheme looks like. In fact, we're actually going to be open sourcing the Pong game that we used and putting it up on GitHub in the next couple of months, so that you can actually start, using our neurons remotely. So what we're actually planning to do is we're actually going to open up our laboratory via the web, so that you can actually write code, new games, for instance. Like, let's say you don't want to play Pong, you want to play Snake. You can make a game of Snake with inputs that define where the space is in an XY position, and you can define what a reward is, like eating an apple, and a punishment as, oh, you went back on yourself or whatever, and write the code and actually deploy it on neurons that we will actually be looking after. So it's kind of like adopt a neuron or whatever, and then you can run your experiments on it. Primarily because we really want people to try it out, right? Like, we'll be doing a lot of this drug testing stuff internally, but then we'll have a couple spare for other people to sort of play around with, regardless of wherever they are in the world.
That's pretty amazing. That's crazy. I'll send out anything, any links that you want, and
Yeah, absolutely. So we're also on Discord. We have team members on Discord as well. So not particularly yet, because we haven't quite launched what we call the NCEP, the Neural Compute Experimentation Platform. But when we do, you'll be able to ask us, hey, how do I do X? Or, oh, we really do need an API for Y or something like that. And we can actually sort of like, so the point is, what we want to do is we want to abstract away all the wetware, all the low level stuff, and give researchers a high level tool that can define what the various electrical stimulus are going to be doing, and what do we do with the return of that information back. That's kind of the goal. And if there are specific requests, like, oh yes, when so we're building a system for V2 where we actually can time events within the game world to actual chemical stimulus. So we are working on things that will drive a syringe, if let's say the Pong ball hits the paddle, we can squirt a bit of caged dopamine, flash a bit of light, and then have that unleash the dopamine. So, timing for drug release and compounds is something that we are working on. So yeah, there's a whole bunch of things in the pipeline. But I think the key for us was to get this paper out and say it is doable. It's not pseudoscience. It's really like a thing, and the data is out there. And if anyone's actually keen or interested, the paper, I think at the bottom, has a link to the OSF, Open Science Foundation, and we've literally just done a whole dump of the data on that. So it's all open, it's all transparent. I think what's his name, Brett, he's done it. So we use mostly Python, so they're in pickled forms. But if anyone really wants, just send me a request. I can actually give them the raw stuff. Having said that, the pickles are probably easier because they're already pre-processed and so forth.
Gotcha. Cool. Thank you so much for talking with us.
No worries. Okay, guys. Have a great rest of the evening, and have a good weekend.
Yeah, you too.
Thank you.
Okay. Bye, guys. Bye.
See ya.